and welcome to my channel, Sam Biology Class. Friends, today we will discuss about another biofertilizer known as azotobacter. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. Azotobacter is a genus of free-living nitrogen-fixing bacteria. It is used in agriculture to promote plant growth, synthesize phytohormones and suppress phytopathogens. It is mass-produced in laboratories in nitrogen-free liquid nutrient media. As a biofertilizer, azotobacter is applied to the field through seed treatment, soil broadcasting and foliar spray, replacing the chemical fertilizer. Azotobacter is aerobic. It normally fixes molecular nitrogen into ammonia without symbiosis. However, some species are associated with plants. Cells of azotobacter are motile, means they have flagella, and they are oval or spherical and form thick-walled cysts. They may also produce a large amount of capsular slime. The cells of azotobacter are free-living. They are gram-negative, found in alkaline and neutral soil. Cells are relatively large, about 2 to 4 micrometer in diameter. Cells are usually oval but may take the shape of rods to spears. In fresh culture, the bacterial cells are mobile due to the presence of numerous flagella. Later, they lose mobility and become almost spherical and produce a thick layer of mucus forming the capsule. Members of this genus contain a full range of enzymes for nitrogen fixation. These enzymes are ferredoxin, hydrogenase and nitrogenase. Azotobacter promote plant growth through a variety of mechanisms. Number 1, it fixes molecular nitrogen into ammonia, thereby making it available to the plants. Number 2, it also synthesizes biologically active substances, for example, some auxins, which stimulate plant growth. Number 3, they promote plant growth indirectly by suppressing phytopathogens. In addition to this bacterial genus is used for bioremediation of soil from heavy metals such as cadmium, mercury and lead. Now the question is how to isolate azotobacter. First step is collection, in which rhizosphere soil sample is collected from certain crops such as wheat, rice, maize or cotton. Second step is cereal dilution, for which 10 gram of soil sample is added to 90 ml of water and is then thoroughly mixed. The resulting suspension is then serially diluted from 10 to power 1 to 10 to power 7. In third step, the diluted suspension is plated on a Jensen's medium and is then incubated at 28 plus or minus 2 degrees centigrade for 48 to 72 hours. This incubation results in the formation of numerous colonies. Next, we have to identify which colonies contain azotobacter. The colonies can be identified by different methods such as macroscopic method, microscopic examination and by chemical test. Macroscopically, the colonies can be identified based on their size. The colonies are usually large, circular, flat, milky white, mucoid and glistening. Second method is microscopic examination. Azotobacters can be identified by gram staining. They are gram negative. Microscopically, the cells are oval, large, sometimes oval to rod shaped, and they often form cysts. Biochemically, azotobacter can be identified by the catalase test, which gives positive result that leads to the release of hydrogen peroxide in air bubbles. Next, what is the process of producing mass inoculum of azotobacter for field application? There are several steps in this process. First step is preparation of starter culture. This culture is prepared by taking suitable amount of pure isolate in 200 ml flask containing nitrogen-free media. The content is thoroughly mixed by placing the flask on a rotary shaker at a speed of 120 revolutions per minute for 48 to 72 hours. Placing the flask on the shaker and operating it at the selected speed results in the separation of azotobacter cells. Second step is mass multiplication, for which the starter culture is transferred to a fermenter containing a suitable amount of nutrient solution. During fermentation, temperature is maintained at 30 degrees centigrade, pH from 7 to 7.2. During this process, there should be continuous aeration. Fermentation results in the formation of a large amount of broth containing very large number of azotobacter cells. Next step is carrier mixing. To apply azotobacter on the field as a biofertilizer, the broth, which has a cell count of 10 to power 9 colony forming unit per ml, is mixed with carrier material such as peat soil, activated charcoal or lignite powder. In addition, calcium carbonate is also added to this mixture to neutralize the pH. Last step is packaging, for which the resulting powder, which contains the azotobacter cells and the carrier material, is packaged in polythene bag. The powder should have a moisture level that is 30 to 40%. The resulting powder can be applied to the field through different methods. First method is seed treatment, in which a slurry is prepared by mixing a suitable amount of azotobacter with water while adding a binder such as jaggery. Seeds to be sown are uniformly coated with the slurry for few minutes, air dried under the shed, and are then sown in the field within 24 hours. Second method is shielding treatment, for which a suspension is prepared by adding azotobacter to water. The roots of the seedlings to be transplanted are dipped in the suspension for few minutes and are then transplanted in the field. Third method is soil application. In this method, azotobacter is mixed in suitable proportion with organic compost or farmyard manure. The resulting mixture is then directly broadcasted on the soil before plowing. In addition, azotobacter can also be applied as a biofertilizer to the plants as foliar spray. So friends, this is all about the isolation, identification, production of mass inoculum and field application of azotobacter. For more updates, subscribe my YouTube channel, Sam Biology Class.